that what you have learned already in biochemistry in last semester, how these things are applied in industrial settings. I mean, generally, that is what we are going to do. And today we are taking, say, basics of biocatalysis, which is one of the things you find in your curriculum. But actually, the basics of biocatalysis is enzymology, which we have studied already. So I'm just going to refer to uh, some of the applications of the enzymes, referring to already what we studied with general biochemistry last semester, and just to see how uh, some of those fundamental principles can be applied in industry. And I think mm, in an hour or maybe even less, some 45 minutes, maybe we should be done, and then you can prepare for the next class so that our classes will not be so continuous like that, like the way we just had this one, 2 hours, and quickly we've come to this one too. All right, so quickly I would say that what we did last semester on enzymes, you have to go back to it and revise it uh, because it will help you to understand the applications that we are going to talk about in industry. And so go back to SBT 401 and revise these things. Looking at um, the energy profile of enzymatic reactions against non-enzymatic reactions. The structure of the enzyme, as we talk about things like the protein parts, or even if it's a catalytic RNA, but mostly it's protein. So we just say the APO enzyme, the prosthetic part, which we refer to as coenzyme, cofactor, active sites, the processes of enzyme activation by these coenzymes and cofactors, and inhibition by some particular substances, which we refer to as inhibitors. Classification of the enzymes, because that's actually the classification naming will tell you what an enzyme can do. And then, of course, the factors that affect this enzyme activity, particularly because you come to see that in industry, it is these factors that we studied: temperature, pH, enzyme and substrate concentration. These are the parameters that are controlled in the digesters or the bioprocessors in the industries. Then you have the, of course, mechanism of action of these enzymes. You want to go and look at these things. Of course, we studied the enzyme kinetics, the Michaelis-Menten equation, and the Michaelis-Menten kinetics, talking about the dependence of the parameters on the dependence of the activity of the enzyme depending on these factors that affect the enzyme's activity. All those kinetic curves that we saw, you want to go revise them to see the dependence of enzyme activity on these parameters. So we take say the dairy industry, for instance, and look at what enzymes are used in this industry and what they are used for. And the first is chymosin, which is also referred to as rennet. And this enzyme is used for milk coagulation in the process of making cheese, for instance. So in the past, rennet, was found to be an enzyme which was in the rumen of these herbivores that when they take in like the young lads of these ruminants, when they take in milk from their mothers, they have the enzyme rennet, which is responsible for the digestion of the milk that they take from their mothers. And so actually in the past, people who were producing cheese, across Europe domestically, were making use of the stomach secretion of the young calves of these ruminants. They just put these extracts, or some actually they take the stomach of uh, the young calf and put the milk into it, or they have the milk and put the stomach into it, or they do extract, sort of some aqueous extract from the stomach of the young lad, and then they put it into the milk for it to cause the curding, which they can then remove. They separate it from the liquid part, the whey, and they can use that to do the cheese. And that's actually it's just the activity of, I think we even saw such a reaction where in the last lecture where you saw that you have enzyme, but only that we looked at it as an enzyme of a bacteria that was acting on the milk protein, casein, and breaking it down into shorter molecules, smaller molecules. And so it's the breakdown of those milk proteins, casein, which results into the formation of the thick curds, the coagulation of the milk, which can then, be used to prepare this, product, cheese. So that's one example of the use of an enzyme in the dairy industry. Now we have other enzymes that are also used in the dairy industry. For instance, we have the lactase, or sometimes you have the beta galactosidase. Of course, again, you see why I said you should revise those things we did. Quickly, when you see the name, you know what it does. The enzyme, breaks down lactose, which is milk sugar, into glucose and galactose. And this is used in the production of milk products for lactose intolerant persons. So you see, the lactose intolerant person is having a deficiency in lactose, in lactase, and because of that, you have the lactose when it gets to the digestive system, particularly going to the large intestine, where it's on its way to get excreted out of the body. Then you have gut bacteria that will be acting on this milk sugar, lactose, and be causing some influx of gas, which will then be resulting into this discomfort and some other reactions in different individuals. But with the introduction of lactase in a milk product, the lactase goes to break down the lactose in the milk, so that issue of lactose intolerance is dealt with, because the product which is being consumed by such a person will no longer be having lactose in it, because the lactose will be broken down by lactase. You have another one, which is lysozyme, which is used for preservation. So instead of putting some synthetic preservatives in the milk products, you rather put some enzyme. That's a more sustainable way of preservation. And this lysozyme, of course, we studied lysozyme. I think we even looked at the mechanism of action of it, breaking down the cell walls, which are made up of peptidoglycan. And this lysozyme, because it causes bactericidal effects, it then can help in the preservation of the milk products, and also prevent, for instance, let's say in the production of cheese, where if you do have some harmful bacteria that or perhaps bacteria that remains in the product after the stage of making the cheese compact, you still have this bacteria that is going to be undergoing some respiratory process, and rather, so when you buy the cheese, you see that the cheese will be having some holes, and big holes, because of the byproduct carbon dioxide that the bacteria will just be releasing into the cheese. And so it causes that what is referred to as late blowing in the cheese. Lipases break down what? Fatty acids. So the lipase will also, hydrolyze milk fat, and this will actually help in enhancing, the development of the cheese. It's a way of breaking down the fats, that are in the cheese. Now, now there's even this sort of engineering of food products, such that you have different percentages of fats in products like this. You also do have another enzyme, which is niacin. Niacin is also an antimicrobial, and is also used for preservation. So you see, it is to inhibit the growth of bacteria, so that the spoilage of the milk products from these pathogenic bacteria is going to be inhibited. So in a way, the shelf life of the product is increased, by the inclusion of this enzyme in the product. Then you can also look at the brewing industry. The brewery industry is there you have production of alcohol, and of course, alcohol mainly is produced from carbohydrates. So you have carbohydrates that undergo some process of fermentation, and in that fermentation process, alcohol is produced. But see, the feedstock which is used, does contain not only digestible, carbohydrates, but can contain also some indigestible carbohydrates, like fibers. And so you do have a mixture of both alpha amylase and beta amylase. You do remember we talked about the specification of alpha amylase and beta amylase, that the alpha amylase breaks down the alpha 1,4 glycosidic bonds, and the beta amylase is capable of breaking down the beta 1,4 glycosidic bonds that the alpha amylase cannot break down. So the combination of these two in a bioprocessor will result into optimal yields from the feedstock that is fed into the bioprocessor. Because if you have just alpha amylase, and the fiber is not able to undergo the fermentation process, because the alpha amylase is not able to break it down, what will happen is that you have less amount of product formed and a lot of waste generated. But once you have a combination of alpha and beta amylase, then you can have, uh, to a maximum effect, the carbohydrates that are present in a feedstock broken down to undergo fermentation to produce the alcohol that is the main product in a product that is being produced, say in the brewery, say in the beer. All right. Now we have also the glucoamylase, which also is used in the brewery. And this particularly is to break down dextrins. And you remember we saw dextrins as also one of those branched polysaccharides that, apart from the 1,4 glycosidic bond, they also have these 1,6 glycosidic bonds. And these 1,6 glycosidic bonds, require another enzyme to break them down. And so that is why we have this glucoamylase. So continuing from the previous slide where we were looking at the beta and the alpha amylase, you have a glucoamylase also in that mixture. So that glucoamylase will also take care of the 1,6 glycosidic bonds that forms in dextrins, so that can also be broken down into glucose and then can undergo the process of fermentation. Now, you see these enzymes we are talking about. So, for instance, when we say alpha amylase, the beta amylase, and glucoamylase, a lot of times these industries have these enzymes already in a mixture that they purchase to make use of. So it's not like they would have to buy each and every one separately. So a lot of times they have formulas that are out there that they can order in the industry. Depending on the particular kind of product that they want to produce, they make use of a particular enzymatic formula which is on the market that they can purchase and make use of. All right. Now, so for instance, see I particularly I've added here that the glucoamylase is used in high gravity brewing to increase the yield of fermentable sugars. So that is if you are adding both alpha amylase, beta amylase, you'll be able to break down only the alpha and beta bonds. How about if you are having some other polysaccharides like the dextrins in there, which are tougher carbohydrates? It means if they are also not broken down, they will just be coming out as waste. And so you have this glucoamylase also in that mixture, helping, specifically with the breaking down of these alpha 1,6 glycosidic bonds. Cellulase. Cellulose is, one of the things commonest of the carbohydrates when we refer to the indigestible part, the fibrous part of carbohydrates. Cellulose is a major part that, again, in the brewery, if you are using alpha amylase, beta amylase, yes, beta amylase can break down those beta 1,6, beta 1,4 glycosidic bonds inside cellulose. But specifically, you have cellulase, which is also specific to the breakdown of cellulose. So yes, you do have cellulase also in some of these enzymatic mixtures that are used in the breweries to break down carbohydrates. Similarly, you have lipase, which breaks down fats into free fatty acids, and that they too can be broken down even the more, and then they can you know get broken down further to undergo fermentation processes. If you go back to the metabolic process of the breakdown of fatty acids, you remember these fatty acids are broken down all the way to acetyl-CoA, and that acetyl-CoA can then go through some fermentation process. And this is to reduce what is referred to as off flavors, which is caused by lipids, fats, and oils in these products such as malts, because if you have these fatty acids in there, they can undergo some sort of oxidation. And sometimes, because you have this, if you have even them broken down into some shorter chains, they can undergo some oxidation, which will be resulting into some, feeling of some. If it was in oil, it would be rancidity, but because it's in beer, it's giving some kind of off flavor, some taste, which is not what is intended. So lipases are also used in the breakdown of free fatty acids. In the breakdown of, say, yes, you break down some. First of all, the lipase breaks down the say if there were some triacylglycerols, you break them down, and then you have some further enzymes which will also break down these free fatty acids into the smaller molecules, which will then go into the process of fermentation. How about the textile industry? So far we've been talking about just food. We first talked about food industry, and then we came to say beverage, which was beer, and now we're looking at what has happened. Is it brewing industry? Okay, I think this is where we were, the textile industry. So the textile industry, there too, you see, you're making use of, things like cotton, uh, so carbohydrate there, you have things like cellulose, and so you need some cellulase uh, that can be applied in what is referred to as, polishing. So in this case, because you're using enzymes, say you are doing some biopolishing, that is to remove what we refer to as microfibrils, some projections out of, say, the fabric which has been produced. So you have, cotton surfaces being smoother, softer. They last more. You reduce some peeling of these fabrics. So you see, I mean, those in textile industry, they understand this more. Textiles with their, different qualities, the ones that are smoother, depending on the kind of polishing that is done to the fabric. You have lacase, which oxidizes phenolic compounds. Phenolic compounds you find, say, in the production of, textiles like denim jeans. So you do have these enzymes that are also applied in the production of such, textiles for, say, biobleaching. In the past, they used some synthetic bleaches in industry for these things, but these days, some of these industries wanting to go green, be more sustainable, be more eco-friendly. They make use of enzymes to do some of these things. The polishing and even dyeing of the fabrics. And of course, this reduces the need for harsh chemicals, which later, get into maybe waste, can get into wastewater, land, things like that. Now you have also pectinase, also will be breaking down pectin. That's another carbohydrate there. Xylinase will also be breaking down. So if you go back to the carbohydrates, this one, I think at the HND level, we did look at different kinds of monosaccharides. When we look at the hexoses, the pentoses, the tetroses, we came across some of these um, monosaccharides like xylose, xylase, and the rest. So these ones come from some of those monosaccharides, which form some polysaccharides like hemicellulose. So pectin, hemicellulose, they are also forms of polysaccharides with some, specific glycosidic bonds in them. That so pectinase will be specific to pectin, and you have xylinase also that will be acting in a breakdown of these glycosidic bonds between the monosaccharides in hemicellulose. All of these enhance fabric quality, and in the process of dyeing, they also help in dye uptake of the fabric. Right, so that is the textile industry. How about the detergent industry? The detergent industry. So if you take your mind to how detergents are used to remove stain. So you have stain usually is having this form of, maybe let's say it's some kind of a lipid. We have some lipid based stains, some kind of oil, and usually you think that, okay, if you want to remove a stain of which is lipid based, you just use some hot water. Maybe a lot of times you soak the material in hot water, and the hot water will help to remove that stain. But if you compare that to making use of an enzyme, lipase, it means that you don't need that high temperature to be able to remove the stain. It's a way of introducing a sustainable, some sustainability, some environmentally friendly process, because once an enzyme, you use lower temperatures, the stain will still be removed. So for instance, you have lipases that can hydrolyze triacylglycerols into free fatty acids, which will be easier to take out of the fabric as stain once you have these free fatty acids and glycerol. You can easily remove, these can easily be removed from the fabric. Monies, so there are also, you do have some of these things that are also based on some polysaccharides, like mannan-based stains, and these two are so these ones you find them in things, so gum, like say gum. This polysaccharides mannan is found in gum from plants. And then we can even say generally, let's say like gum from the gum that you chew, for instance, is if it gets into a fabric that is stained, that its removal actually can also be by this enzyme. So what it is that you have these detergents that are incorporated with these enzymes. So the enzymes are like ingredients in the detergent you buy, and it's in it, And so you have these eco-friendly detergents which are having these enzymes in them, which sort of improves their uh, you know effectiveness in the removal of stains from the fabrics when they are being used. And at the same time, you will not need to use high temperatures to be able to remove these stains again. So another one is pectinase. I think we saw pectinase already, but this time around, particularly in the detergent industry, we see pectinase was being used in the brewery industry, for instance. And here again, you are seeing it being used in detergents because you have some pectin-based stains also. That's, pectinase can help to hydrolyze and remove them out of, fabrics. Same way, lacase we saw, which was being used in the processing of fabrics. And here we see it because it can oxidize phenols. We have a lot of phenols in products like wine, tea, coffee. So those stains that come from wine, tea, coffee, cocoa drinks, things like that. You have licases that when they are infused into the detergents, they help in the removal of those stains. So usually it's not like you have only a particular enzyme which is infused in one detergent. You actually have a number of them so that they can all act depending on the stain which is available. The washing of the detergent will be improved by the presence of these enzymes that are in the detergent. The biofuel industry, I think I mentioned the last time, the biggest biofuel industry you find is in Brazil, that they produce a lot of this bioethanol from sugar cane. And sugar cane is what is having sucrose in it. And so it's there too is a process of fermentation, which is resulting into the production of this bioethanol. And you have enzymes such as phytase, which can, hydrolyze, some other component of these polysaccharides as phytic acid. So that way you have, say, phytase. If you put all of these phytase, glucosidase, hemicellulase, xylanase, what it does is, the same effect that we talked about in the production of ethanol in a brewery, that each of these enzymes are going to be acting on some particular polysaccharide that is having some specific glycosidic bonds, that if one enzyme is not able to break it down, the other can break it down. So that if in the production of biofuel there is amylase, like amylase is the primary base, the primary enzyme, which is there, to cause the breakdown of these polysaccharides to further undergo the fermentation process. You then will have these other enzymes as a secondary enzymes to break down, like fibers that are in there. That's the undigestible parts, different forms like hemicellulose can be broken down by hemicellulase. Xylan will be broken down by xylanase. And you have glucoside, beta glucosidase can break down, say, cellobiose. So different, polysaccharides that are present in there can be broken down. In that way, you have the production of, the ethanol being improved. If this is not done, there will be a lot of waste that will be generated. A lot of the carbohydrate cannot be broken down to go into the process of fermentation. And that is not going to be economical if you are producing a lot of waste and the product which you need, which is the biofuel, you are not producing it in large amounts that you should, then you'll be running at a loss in the end. I mean, it's just like, this reminds me of some time ago, I think during the time of President Kufuor, he tried to, introduce this cassava processing into, different products like starch. I think they produce some cassava chips, things like that for different things. But it wasn't so sustainable. I think that thing has died off. There hasn't been so much of the uh, development of cassava products in our country, except for gari, which is also not really at an industrial scale per se, because you only find women in some of our, towns and villages and that fry this gari and pack it in sacks. And then you have some people who go buy from them. Some people actually buy to export out of the country and things like that. But that's just about gari. But the other things which President Kufuor tried to, let cassava be used to produce like starch for the textile industry and things like that, hasn't really been improved so much. Except these days, you also hear some of these breweries are making use of cassava to produce some beers, and that's it. But in the world, the country with the best production of cassava is Thailand. You see, these things have the basis. They have some scientific base. You need to do some serious scientific research to have the best yield out of these things. So, for instance, why is Thailand, the biggest in the production of cassava and its products? Because they have been able to engineer and have, a variety of cassava that produces like, the highest yield of carbohydrate. Uh, so the highest yield of not carbohydrate, the highest yield of starch. So if you have cassava that's yeah, I think the cassava we have in Ghana is producing a starch yield of about between 50 to 60% or so, and you have Thailand having a cassava that is in the 90%. I mean, you can't compete with that. You see, so, and much of our cassava too, we grow it and we just want to have some fufu and have some banku out of it, and that is it. But industrially, if you want to use it industrially, then we have to study it much more scientifically and come up with the variety that gives us the highest yield. So for instance, if we say we want to make use of cassava to produce bioethanol, for instance, uh, first you have to get cassava which you are using for food to sustain the whole population, and then you have the industrial cassava, which you make sure that cassava you scientifically study it, improve it, and make sure it gives you the highest yield possible of, say, the starch which you need for the production of, say, the bioethanol from it. So, I mean, you can't underplay scientific research. You can't develop seriously without scientific research. You need to study things and get the best optimal system in before you will be able to actually have some economic advantage. All right. Now you have uh, some animal sources of. Let's take a look at the sources of these industrial enzymes. You can have some of these enzymes, obviously, coming from animals. Like for instance, you have trypsin, uh, which actually we saw as an enzyme which can be used in the leather industry, in the meat industry as a meat tenderizer. You remember, and even in pharmaceutical industry, yes, it's used in some pharmaceutical products as for debridement, the treatment of wounds. You have it there that you have pancreatin that can come from an animal. So see all these enzyme trypsin, pancreatin, pepsin, collagenase, they come from animal sources, except, okay, for this last one, collagenase, we can say it's coming from some microbial source. But you see all the rest, trypsin, you can get it from cattle, pigs. That is originally, it was being gotten from this, originally the production of these things were like, for instance, what we saw with the production of cheese, the young cows. The young cows were actually being killed, and their stomachs, and the rennet or the chymosin was gotten from their stomachs. I mean, things also like the pancreatin. But these days, because of scientific advancement, or you can synthesize these things. I mean, just by culturing, you can, these enzymes are synthesized, not say chemically in a lab, but what happens is you have industries actually that are specialized in the production of enzymes, so that they only take enzyme, some tissue that once it's cultured, you can give it some optimal conditions like temperature, pH. In some bioprocessor, biodigester, and you have, for instance, if it's a microorganism that produces some of these enzymes, they will produce more of it. And then it's protein. So it's like an extraction. Once you extract it, what you do is you then take it through some purification processes. So you actually have these enzymes on the market that are being sold. If you are in this industry, you look for these enzymes. They are there. So you saw even in the first slide, when you saw the chymosin slide, you saw that you had the chymosin or rennet in a bottle that is being sold. So the food industry people, they have these things that they buy and make use of. Similarly, if you are in the textile industry and you want to make use of these enzymes in any of your processes, these things are on the market. You order them, you have them, and they are even in different specifications. And then you use them. Those in the brewery industry, they also have some of these enzymes that they make use of as different formulas depending on the products that they want to come up with. Right. Continuing with the animal sources. So I've talked about this chymosin already, which I said in the past, you got it from the young cows. But of course now the industries actually we have industries that produce enzymes. So we have industries that their mandate is to produce the enzymes, and so they produce the enzymes, and other industries also get these enzymes and make use of them in their industries. So you see, one industry depends on the other. That's how it is. So you have this elastase, lipase, urokinase, all of these something like urokinase like this, I mean, originally in a crude form was being getting from dog urine. Because look at the name, urokinase. It's in urine. Of course, now is you can just produce this industrially and then make it available in bottles. People will buy and make use of it. All right. Microbial sources. Yes, a lot of enzymes are produced from microbes, which are like, some little factories. Like if you take a microbial cell, that microbial cell has, metabolic processes that it is undergoing. And so it's like you just have to give the microbe a condition in which it will grow and produce the enzyme that you want, and then you harvest the enzyme. I think I had some. I will look for some video link. I hope I can have access to such a video link again, and then you can watch some of these processes of production of enzymes in industries, which you can relate to as some form of virtual excursion into. There are some short videos, not long, and then you see how these enzymes are being produced in different industries. Like this, bacillus species, will is used to produce, like, this common enzyme we're talking about, amylase, protease, cellulase. You can have, bacteria producing that for you. Another bacteria, a Trichoderma rhizae, is well, this is not bacteria. This is a fungus, and that produces cellulase, hemicellulase. Yeast is a common one which is used in bakery and also in brewing, which produces the enzyme invertase and lipase. Invertase breaks down sucrose, into glucose and fructose. Lipase, you already know, breaks down lipids. You have another fungus, Aspergillus niger, also producing some amylase, glucoamylase, pectinase, phytase. So you see what I was saying, that these enzymes are being produced by some industries for other industries, and a lot of times is this bacteria they use. You only first prepare a culture of these microorganism, and then you introduce that microorganism into a bioprocessor, which is a larger environment, larger container, and those factors that we talked about, pH, temperature, concentration of the substrate, that is the feedstock, what you put in there for the bacteria to feed on, and out of it, because of it undergoing that metabolic process, will be producing these enzymes for you. Remember, these enzymes are proteins, and so that's like some protein synthesis, protein synthesis, which is ongoing in the bacteria, and is either the enzyme, is that protein is being produced intracellular in the cell, so that later there are some other sophisticated processes by which the cells are broken, they are ruptured, so that the enzyme is now harvested from the mixture of the fragments of the cell, and then you have a separation, purification of this protein from that mixture, or the microorganism is secreting these enzymes as an extracellular product. So if it's an extracellular product, it will not require any sort of rupturing of the cell because it's already in the extracellular space. So you only have to take the fluid and just extract the enzyme, which is the protein, out of it, purify it, and then you can make use of it. Plant sources. So we did talk about the enzyme papain, which is used in meat tenderizers. It's also used in pharmaceutical products, and that papain can be gotten from, say, pawpaw. You have others, say amylase can be gotten from, say, barley. That's a grain. You have an enzyme, bromelain, can be gotten from pineapple. Also used in meat tenderizers. It also has that effect, that digestive effect on meat. And you do have, which is why sometimes some people make use of this, say, pineapple juice, to um, soften meat, for instance. And you have apples, oranges, grapes. They also have this enzyme, pectinase. So these are some fruits, and these are also applied in, say, the wine industry, even in the textile industry. You have these enzymes, uh, you know, gotten from these fruits, which are used in those industries. All right. More plants, the castor beans, Ricinus communis, has this lipase. Of course, many of these plants have, lots of lipases because, of course, you do remember that these both plants and animal cells, have a lot of these, in the cell membrane, you have a bilipid layer. But even apart from that, within the plants themselves, they have these enzymes, lipases, which they also use in their metabolic processes, which can be extracted and utilized in different industries. Like for instance, these lipases are not used only in the food industry. They are also used in detergent, because we just saw that lipases can be used in detergents. Lipases can be used in say, the brewery, same way as in the dairy industry. So whilst you're using a lipase in the dairy industry to, improve flavor here, same way in the brewery, lipases are also used to improve flavor, because it's to prevent some off flavors in beers. And at the same time, in detergents, to remove fats, or like to the lipid-based stains. And at the same time, in the production of biofuel, you still, make use of these lipases. You do see another one, phytase, in soybeans, which is also used, in enhancing, what is being referred to as bioavailability of phosphorus and other nutrients, because, the plant is known to be involved in like nitrogen and phosphorus fixation and uptake from the soil. So the enzyme which is in that plant, which helps in the uptake of phosphorus from the soil, if it is applied, it then makes available, more of these, phosphorus as nutrients for plants. Some kind of call it biofertilization in agriculture. You do have these sugar beets and sugar cane also producing invertase Of course, these plants, they store carbohydrates in their roots that they make use of for food. So they require this enzyme for the breakdown of say sucrose. And so they produce a lot of this enzyme. Lactase from another plant, the Japanese lacca tree. So that's another source of another plant source of enzymes, which is used in bioremediation, textile dyeing, and even in the paper industry. That is used in paper processing, in the breakdown of the another undigested carbohydrate, lignin, sort of making paper smoother. You use lactase in there, and that can be gotten from the plant, the Japanese lacca tree. All right, so I think we've done well. I mean, 5 minutes after uh, 3 o'clock. No, 3 o'clock here, but with you, that's going to be I think 1 o'clock. No, 12 o'clock. So you can have some enough break before you start the next lecture. Any questions, any contributions? The floor is open. I will listen to you. You can raise your hand. I will enable your microphone. Then I call your name, you speak, or you can write in the chat. All right. So the floor is open.